via filtration. Okay, so there are 3 steps. Remember when we talked about respiration, we had 3 phases? We had pulmonary ventilation, and then we had external respiration, which is movement of air in and out of the airways, and external respiration, which was gas exchange across the alveoli to the blood and blood to the alveoli. And then we had internal respiration, which was the gas exchange from the capillaries to the tissues. Okay? So when we talk about urine formation, we have 3 basic components that actually change the content of the filtrate. All right? So within the nephron, and the nephron, remember, starts with the capsule, glomerular capsule or Bowman's capsule. And then it forms the proximal convoluted tubule. And we have the descending limb of the nephron loop, or you could call it the loop of Henle. And this has both thick and thin segments. And then we have the ascending limb, and then it has thin, which then changes to thick. We have the distal tubule, which soon becomes the convoluted tubule. And that's the nephron, okay? And the distal tubule is going to empty into the collecting duct. And then multiple collecting ducts become papillary ducts. And at that point, they empty via the papillary ducts into the minor calyx. That was all that we talked about on Thursday, all right? So at the Bowman's capsule, we have the content of the blood becoming filtered, and so the entire content of the nephron is identified as filtrate. So once small particles, including water, are removed from the blood and enter the Bowman's capsule, it's known as filtrate. And all the modifications that occur through the nephron, it's still filtrate. All the modifications that occur in the collecting duct, it's still filtrate. And we don't change it to urine until it actually drips into the minor calyx. Okay? So when we talk about urine formation, we have the events that occur in the capsule, one final event that occurs in the collecting duct. So we're going to finish up by talking about what occurs in the collecting duct today, just very briefly, not even the cells of the collecting duct, only in reference to what the nephron loop does. So we're going to get to this point today, and then on Thursday we're going to start with this and look at the role of the collecting duct. Okay? So the 3 stages by which urine is formed is, first of all, the formation of filtrate. Okay? So that occurs across the glomerular capsule. Glomerular filtration. We don't have any other kind of filtration. All right? So it's not like there's tubular filtration or anything else. Filtration occurs at the capsule. In the proximal convoluted tubule, we do reabsorption and secretion. And then again at the distal convoluted tubule, we do reabsorption and secretion. This is going to modify the content. Remember the story I told about putting all the little things in the box? Okay, that's the filtration. Anything that fits in the box, anything that passes the filtration membrane becomes part of the filtrate. And then we reabsorb or secrete purely by amount, concentration. Okay, so this is non-regulated. This is regulated. And that leaves us with our nephron loop. Okay? The nephron loop, which is what we're going to finish up with today, well, functions to form an osmotic gradient. And that's all I'm going to say about it until we talk about it after break. Okay? We'll have about a short lecture after break. So its function is to form the concentration gradient, and then the concentration actually takes place in the collecting duct. So the filtrate is concentrated, if it needs to be, in the collecting duct. Obviously, if you drank 2 gallons of water before you came to class today, you're not going to need to concentrate your urine, okay? Your filtrate into urine, and so that won't take place. You'll pee out a very dilute, very light-colored urine. But if it needs to take place, that's where it takes place. So, that's kind of an overview of what we're going to talk about today. Going from the Bowman's capsule through the nephron loop, and then we'll start over again with the nephron loop on Thursday and go through the distal tubule. All right? So let's look at the capsule. And I'm going to kind of draw part of this. So can I erase what's on the board now? You guys okay with that?
Yeah.
So what's our definition from Bio 430 of the process of filtration? What does that mean? Real basic sentence.
Separating smaller from larger particles.
Okay, it's separation of particles based on size. And so that we have a membrane across which this occurs, and it's formed by a sandwich of simple squamous epithelia. So remember our respiratory membrane, not the respiratory epithelium, but the respiratory membrane was the simple squamous epithelium of the alveolus, connective tissue. There's some peanut butter in there, and then simple squamous epithelium of the capillary. And that was the wall that the gases had to cross back and forth. All right? So we have blood flowing into the capsule via the afferent arteriole. It loses its smooth muscle and becomes a glomerular capillary. And then the small particles have to cross the wall, the simple squamous epithelium of that capillary. They have to pass the wall formed by the inner or visceral layer of our capsule. And then finally, it's those particles and fluid are within the capsular space. Okay? So let's take a look at what that filtration membrane is formed of. So I'm going to draw first the simple squamous epithelium of the capillary. And we'll draw both sides of the capillary here. So this is not the peritubular capillary, this is the glomerular capillary. And simple squamous epithelium. every epithelium has what structure just deep to it?
Basal layer.
A basement membrane, okay, the basal lamina and the reticular lamina. So we'll draw that basement membrane in here. It's a little thicker appearing when we see it on the electron micrograph. And then we have the epithelial layer of our capsular wall, which is also simple squamous epithelia, all right? But it's a unique, it's these components right here. It looks almost like a dinosaur or something with these scales on it. But I'm just going to draw it on one side. It would be on the other side here as well. But we have this cell where the nucleus is found. And then it sends down foot-like processes known as pedicels. Since it's an epithelium, it also has a basement membrane. So now we have this double basement membrane. No connective tissue, just this double basement membrane. And this special epithelial cell that's covering the capillary and wrapping around it is known as a podocyte. So that is formed of the cells that form the visceral layer of the Bowman's capsule. Now we're not quite done yet because we have multiple podocytes. So I'm going to draw the next podocyte in purple just so that you can distinguish it a bit. And it also has these foot-like processes. And each foot-like process interdigitates with the pedicel or foot-like process of another cell. So every other pedicel belongs to a different cell. And these foot-like processes, again, are known as pedicels. So the cell is the podocyte, and these structures sticking outward are the pedicels. All right? So we'd have the same thing on the reverse side of our capillary wall. I'm not going to label it, but I'll just draw it in there. Okay? So that is our filtration membrane. The podocyte, two basement membranes, and the capillary simple squamous epithelium. Yes?
But they're all, the purple and the blue cells are both podocytes.
They're both podocytes. I just drew one purple so that you could tell that every other pedicel belonged to the same cell. Okay? All right, so now, what structures? Remember, this is our filtration membrane. So this is going to contain the plasma of our blood. Our our red blood cells and white blood cells, all right? And if our blood is filtered across, then the fluid out here is going to be our filtrate. In other words, the filtrate is going to be out here in our capsular space. All right, so the blood's in our capillary, and we push the fluid and small particles out, and it enters into the capsular space. So if I were going to really define out here, I would have the parietal capsular layer forming the outermost boundary. But we're not quite done yet looking at the filtration components. Because remember, it's based on size, so we have to determine some component of size. So here's a smaller scanning electron micrograph of the podocytes, and then these interdigitating pedicels. All right? And if we look at that in a scanning electron micrograph, here would be the podocyte. Here would be its pedicels coming down. Every other one being, so here's this one for this cell, here's another one for this cell. You can see there's a different one in between. So it's every other. And here would be the capillary, simple squamous epithelium. So blood versus filtrate. Down here is this layer enlarged. Okay. So here's the simple squamous epithelium. Notice that there are openings in the simple squamous epithelium of the capillary membrane. So let me move that up a little bit higher for you guys. So here's the capillary with these openings. Here's the two layers of basement membrane. And here are the pedicels. Each of these belongs to the same cell. Each of these belongs to a different cell. All right? And so that is the structure of the filtration membrane. So I'm going to take what I have there and draw it larger like this so we can label the parts. Because this is what creates, if we have filter paper, I'm going to erase this part here so I can use it for the diagram. If we have filter paper, there's spaces between the fibers, and the size of those spaces determines what can pass through the filter paper. So here's a pedicel of one podocyte, and here's the interdigitating pedicels of another podocyte. And there's a gap between the two. All right? There's actually a membrane that crosses that gap. So this is known as a diaphragm. But the gap itself is known as a filtration or glomerular slit. So that's what we have here. So some particle that's going to pass through there has to be small enough to pass between the spaces of each of these pedicels. And then we have our basement membrane. Two layers. And then we have our simple squamous epithelial cell. But it has openings. Remember when we talked about capillaries, we had fenestrated capillaries? That's what these are. So if somebody took a hole punch, one of those single hole punched, and punched a hole through a fried egg, and it punched that hole, the edges of the membrane sealed so the cytoplasm didn't leak out. and we have this channel that passes right through the cell. So these openings here are the fenestra, or the pores. So that's what something has to pass through if it's going to leave the blood and enter as filtrate. Particles have to be small enough to do three things. They have to be small enough to pass through the fenestra. They have to pass among the collagen proteins, the different proteins of the basement membrane. And that's probably the most difficult, restrictive part. Okay, charges are also involved. And then they have to pass between the slits. Finally, they're in the capsular space. Okay? So that's the process of filtration. Now, diseases can affect this, all right? So, inflammation of the glomeruli can be caused by a streptococcal, usually, sometimes staphylococcal infection, where the bacteria release toxins. And remember the process of neutralization that we talked about with antibodies, where the toxins are bound to antibodies that cover them and don't allow them to be taken up by cells or to enter into cells and cause their deleterious effects. Well, that antibody toxin complex, it's stuck in this basement membrane and in the pores and can block the filtration process. So what it does is it increases the filtration pressure elsewhere and causes all of these to spread apart. And the pores open up and the filtration slits spread apart, and we can end up with large particles such as proteins in the filtrate that would normally wouldn't be present there. Okay? That can also happen transiently if someone is exercising really, really hard. So when my husband was training for his first marathon, actually the only one he ran, he did 2 other marathons as part of a relay, but the only one he ran by himself. He was doing several, miles every day, and then on the weekends he'd run 15 to 18 miles, kind of building up for the marathon. And one morning he called me from LA. He called me about 3 o'clock in the morning. And he said, I'm not feeling very well. And he said, I think I have a fever and low back pain. And I said, with the low back pain, it sounds like a kidney infection. And he had run quite a bit the day before and hadn't drunk very much fluid. And so it's pretty unusual for guys to get kidney infections because of the long urethra, whereas women have shorter ones. And so it's much shorter distance for bacteria to travel up to the bladder. But it was partly because of that intense exercise and all the protein that he had broken down from his muscles and doing that exercise that ended up causing that. And he ended up driving himself to the hospital, and they put him on IVs for 3 hours. And then he said, well, I've got a meeting at 8 I've got to go to. So he got up to get dressed and promptly fainted on the floor. So they kept him a little bit longer. But this is what we mean by the filtration membrane, okay? The 2 epithelia with the basement membrane in between. So, what is the force then that is required for filtration? We're talking about diffusion. The things that move particles in diffusion is the random bumping around each other, all right? And that if you have a lot of particles in a small space and they're randomly moving around, that energy is increased with temperature and concentration, and more likely to bump into each other, and that causes them to move apart. All right, so that's a real general description of diffusion. But that doesn't work for filtration. All right, we have to have some kind of force pushing these particles through the small spaces. So where does that force come from?
Blood pressure.
Blood pressure. All right, if we're looking at a piece of filter paper, the gravitational force is going to pull the fluid through the filter paper, and the particles that are small enough will pass through. If we can even in a chemistry lab put a vacuum tube on it and run water, and that creates a vacuum tube that increases the force of filtration. But what we have in the body is blood pressure, all right? Can I erase what's on the board now? You guys done with that? I want to kind of redraw what's over here. and I'll leave part of this diagram on here. So glomerular filtration pressure is the result of opposing forces. So the highest pressure is going to be that force of the blood pushing against the vascular, the capillary walls. Remember the diagram that I drew when we did the lymphatic system that looked like this? When we talked about the formation of lymphatic fluid? And we talked about hydrostatic pressure as a pushing pressure. What was the opposing force?
Osmotic pressure.
Osmotic pressure. And that was due to the proteins that we find in the blood. And that was going to keep fluid. So on the arterial end, we had this relationship, and on the venous end, we have this relationship. All right, so hydrostatic pressure, capillary hydrostatic pressure, is going to be the force pushing particles and water molecules out of across the fenestra, in between the slits, and into the capsular space. Normally, under normal situations, normal blood pressure, this capillary pressure, capillary hydrostatic pressure, is equal to 50 millimeters of mercury. Okay? A little high for most capillaries, but remember our efferent arterial is normally smaller in diameter, and that's going to cause blood to back up into the glomerular capillaries and raise our capillary pressure. All right? But we also have proteins in blood. Remember what the major osmotic protein in blood is? Albumin. So albumin is going to create an opposing osmotic pressure. So that is what's called here as the colloid osmotic pressure. Okay? I'm just going to say capillary. And that's about 30 millimeters, okay, as an opposing pressure. So if that's all we were dealing with, we would be left with the net 20 millimeters of pressure pushing particles and fluid across our filtration membrane. But as that fluid is pushed across, so we end up really with a net flow. It goes into the capsular space, and on that side, that volume of fluid is going to create its own hydrostatic pressure. Okay? Even though there's no heart contracting, the very fact that there's a fluid there is going to oppose more fluid coming in. And so that is identified as the capsular hydrostatic pressure. And that's going to push back. So that's going to have a force pushing back on this fluid, resisting additional fluid coming in. All right? Now, that's about 10 millimeters of mercury on the pushback. Does the capsular space, does the filtrate have any proteins in it to create an osmotic pressure that's going to pull additional fluid into the capsular space? Pardon? It shouldn't, unless the filtration membrane is damaged. We don't have proteins, amino acids, but we don't have proteins in the filtrate. All all right? They're too large to cross the filtration membrane. So that leaves us then with a normal glomerular filtration pressure of 10 millimeters of mercury. Okay. Once we subtract the opposing pressures of the proteins in the blood versus the fluid that's on the other side, already in the capsular space. So let's say that our capsular hydrostatic pressure drops. If it drops by just a few millimeters, let's say it drops from 50 millimeters of mercury to 45 millimeters of mercury, and nothing else changes, that drops our glomerular filtration pressure in half, just losing 5 millimeters of mercury of pressure within the capillaries. Now probably if we drop that filtration pressure, the amount of fluid in our capsular space will drop as well. So this number might be lower. But it doesn't take much of a drop of the hydrostatic pressure to radically change the net glomerular filtration pressure. Is there a question?
Yeah, in the capillaries, the CHS, is it?
This is the capillary hydrostatic pressure. The red, the blood pushing against the capillary wall, okay? So as we increase blood volume in the body, or we increase cardiac output, what's going to happen to capillary hydrostatic pressure if nothing else changes? It's going to go up, okay? And if we lose blood volume, dehydrated, or hemorrhaging, then capillary, and nothing else changes, then capillary hydrostatic pressure goes down, okay? So this means that it's very important for the kidney function to regulate blood pressure, all right? And so we're going to look at the site of the afferent, efferent arterioles and the distal tubule as the primary location where this is regulated. If I go back to a couple of pictures previously where we looked at the capsule, here we have a unique arrangement where, remember the distal convoluted tubule, the ascending limb, and then I said there was a short section of just the distal tubule before it becomes convoluted. That distal tubule comes up here and is found between the afferent and the efferent arterioles. Now this is drawn anatomically correct. In our models, here's the proximal convoluted tubule over here. In the models, they'll have the distal tubule about this big, which is way too small. Okay, I think they were just trying to save plastic or something. But we have two sets of cells that are important in providing an increased pressure within the capillaries if the rest of the systemic pressure drops. Because if it drops too much, we lose our filtration pressure. Okay? And this apparatus is known as the juxtaglomerular apparatus. So I'm going to leave this image up on the screen while I draw it on the board and we look at its function. Can I erase what's on the board now? Okay. So there are two ways by which we can modify, two simplistic ways by which we can modify or elevate the glomerular filtration pressure. Okay, so diagrammatically, if we look at this very small diagram, we have a wide afferent arteriole forming our glomerular capillaries, and a smaller efferent arteriole carrying blood away. This is the normal pattern. So afferent and efferent. That in itself increases capillary hydrostatic pressure, right? Because we have a smaller outflow, smaller tube for the fluid to pass to leave. So, if pressure drops, okay, if blood pressure drops, what can we do to change the pressure within the capillary? Constrict this further and dilate this further, okay? So we can dilate the afferent arterial, constrict the efferent arterial. Okay, right, that's a very local and quick mechanism by which the kidneys can respond, and that's called an autoregulation. It doesn't require systemic changes. What else can we do? Obviously, we can constrict systemic vessels, okay? So this is a local autoregulation. On a more systemic effect, we can constrict peripheral arterioles in the body. That's going to increase peripheral resistance and therefore increase blood pressure, okay? What else can we do on a systemic level if we want to increase blood pressure? Thinking back to our lecture on blood pressure and cardiac output. What can we do to increase blood pressure?
Increase contractile force.
Yes, we can do that.
Cardiac output.
Increase, which was another way of increasing cardiac output. How do we increase venous return? And it, besides changing cardiac output.
Osmotic.
No. Well, we can change osmotic pressure to end up with that. So how do we increase venous return? We can exercise, do all sorts of things, but what's venous return? Volume. How can we increase cardiac output? Increase overall blood volume, okay? So one of the ways in which we can do this systemic aspect is to increase overall blood volume. Right, so with that in mind, we can look at how the juxtaglomerular apparatus actually works, okay? So it has two components. One being the muscle cells of the arteriole, and other component being the specialized cells of this distal tubule. So here's the afferent arteriole. 2 layers of smooth muscle. And in a specific region of the afferent arteriole, just as it comes into the glomerulus, these smooth muscle cells have special molecules, enzymatic molecules known as renin. So these smooth muscle cells are known as juxta, which means next to, juxtaglomerular cells, or because of the granules of renin, granular cells. Okay. That's one part of our juxtaglomerular apparatus. Question?
Did you say that they were the smooth muscle cells?
They're smooth muscle. the smooth muscle of the afferent arteriole. And then, of course, this becomes our glomerulus. And then leaves as our efferent arteriole. And between the two, we have a segment of the distal tubule. Which is formed of simple cuboidal cells, except for the region facing the capsule in the glomerulus. And these are slender, narrow, simple columnar cells. Okay, it's the only time in the nephron that we actually have simple columnar cells. The rest of the nephron is formed of an alternating simple squamous in our capsule, simple cuboidal in the proximal convoluted tubule, simple squamous in the nephron loop, simple cuboidal in the distal convoluted tubule. So in this specific region right here, facing the capsule and the glomerulus, we have this narrow region. Because it's narrow, the nuclei are very close together. And so this is identified as the macula densa. The rest are just the normal simple cuboidal cells that we have in the distal tubule. So the macula densa is the second component of our juxtaglomerular apparatus. So juxtaglomerular cells of the afferent arteriole, macula densa of our distal tubule together work to respond to low blood pressure within the capsule. So let's see how that works.
Wait, so the macula densa is the columnar cells?
Yes,
Or-
because macula means spot and densa refers to dense spot. So it's all those nuclei close together forms a dense spot, and that's where that term comes from, okay? So it's not the entire distal tubule, it's just that one segment of it right where it passes between the afferent and efferent arterioles. All right, so let's see how this works. And some of this we've already covered very briefly with the blood pressure. We just learned about certain hormones. We didn't learn about this whole process. So I'm going to erase this portion over here, okay, and go through in a little bit more detail how it occurs.
The efferent arteriole is the same juxtaglomerular.
The efferent arteriole does not have juxtaglomerular cells. That's only in the afferent arteriole. Okay, did you guys hear that? So JG cells, the juxtaglomerular cells are not found in the efferent arteriole.
And what composes of the,
Pardon?
and what makes that?
Just smooth muscle, just a typical smooth muscle, but without those components. All right, so if we have a decrease in blood volume, all right, or for some reason we have low pressure of blood passing through the afferent arteriole that's not stretching out the afferent arterial walls, okay? That is detected by these smooth muscle cells, and they respond by releasing renin, all right? There are three things that can cause the JG cells to release renin. So JG cells release renin due to one, relaxation of afferent arterial diameter. come back and talk about what actually happens, what the renin actually does. Okay? Another mechanism that causes the renin to be released is signaling from the macula densa. So let me explain that really briefly. We'll look at the next list. We'll come back to this one. All right, the macula densa, that special part of the distal tubule, has osmoreceptors. Osmoreceptors mean they're sensitive to the particle concentration. We have those in the hypothalamus. So if you don't drink very much, the concentration of sodium ions in your blood goes up, and those osmoreceptors in the blood vessels of your hypothalamus sense that and trigger a sense of thirst in your body. So you're driven to go get a drink, all right, and add fluid to your body and dilute out those sodium concentrations. So in the macula densa, these are sensitive to the level, the number of sodium ions present in the filtrate. Well, what does that mean? We haven't talked about the role of the proximal convoluted tubule yet, so let me just move ahead to this picture. Here's our capsule. Filtration of sodium ions means that the sodium goes into the filtrate, and then it goes into the proximal convoluted tubule. There, 80% of the sodium ions are moved out of the filtrate back into the blood. If pressure is high, if glomerular filtration pressure is high, what is that going to do to the rate at which this filtrate flows through the nephron? Is it going to increase the rate or decrease the rate? So if filtration pressure is high, and we're pushing lots of fluid into our capsular space, and that fluid then goes into the proximal convoluted tubule, is it going to flow through the proximal convoluted tubule at a faster rate or slower rate than if our filtration was decreased? At a faster rate, okay? So, next concept, if it goes through at a fast rate, are the sodium ions going to have very much time to be taken out of the convoluted tubule and added back to the capillaries? No. So we're going to end up with a higher number of sodium ions in our nephron by the time it comes back through the distal tubule. Okay? Well, our concern is if our filtration rate is low because of a low blood volume. So, if filtration rate is low because our pressure is low due to low blood pressure, then as it gets into the proximal convoluted tubule, it's going to flow more slowly. Hence, more time for sodium to be moved out of the filtrate and back to the blood. So when it gets back via the distal tubules up here into our macula densa, there's going to be a decrease in the number of sodium ions available in the solution. Okay? So if filtration pressure is low, fewer sodium ions will be present in the filtrate. And that is picked up by the macula densa. Okay? So the macula densa cells basically are osmoreceptors for the amount of sodium in the distal tubule. Does that make sense? Why? Okay? And they will release prostaglandins, paracrine hormones, if you will, because they don't get in the blood, they just travel a short distance over here.